Welcome to the Deep Dive. Today we're tackling something quite complex, sparked by cases like the Karen Reed and Brian Kohberger trials.
Right. The big question floating around is whether these cases maybe hint at DNA evidence being used improperly sometimes.
Exactly. Could law enforcement or the FBI misuse DNA, maybe even leading to, well, convicting innocent people?
Yeah.
It feels like a crucial question to explore.
It absolutely is. So in this deep dive, our goal is really to unpack that. We'll look at how DNA evidence is actually used, in criminal
And
investigations.
how reliable is it, fundamentally?
Yes, assess its reliability, but also look at times when maybe the interpretation went wrong or the circumstances around it raised some red flags. We need that background first.
Okay, so DNA. It seems like this almost magical tool now in solving crimes. How does it actually work? How do they go from, say, a tiny sample to identifying a person?
Well, forensic DNA analysis has really exploded since the late 80s. Initially, the main technique involved something called short tandem repeats, or STRs.
STRs. Okay.
Think of them as like really specific markers, almost like a genetic fingerprint that's unique to each person. More recently, SNPs have become important too.
SMPs.
Single nucleotide polymorphisms. Basically, variations at single points in your DNA. They're especially useful when the DNA is maybe degraded or you're doing more complex searching.
And these profiles, these STRs and SNPs, they get compared against databases, right? How does that whole system work?
That's right. Law enforcement primarily uses CODIS, the Combined DNA Index System. That mostly holds STR profiles, typically from people convicted of certain crimes or from crime scenes themselves.
Okay, CODIS.
But beyond CODIS, there's been this huge development using commercial third-party DNA databases. This is where investigative forensic genetic genealogy, or IFGG, comes in.
IFTG. That sounds interesting. Maybe a little controversial, too. How's that different from just searching CODIS?
It's definitely a different approach. It's more indirect. You see, when they don't find a direct match using STR and CODIS,
Which probably happens quite a bit.
it can, yes. So with IFTG, they analyze the SNP data from the crime scene sample and upload it to these commercial genealogy sites, the ones people use to trace their ancestry.
Oh, right, like Ancestry DNA or 23andMe, those kinds of places. Though maybe not the specific ones, depending on their policies.
Exactly, those kinds of databases, though yes, policies vary. The goal isn't to find the actual perpetrator directly in that database, it's to find relatives. Distant cousins sometimes.
Ah, so it's like casting a wider net to find the family tree and then sort of zeroing in from there.
Precisely. Genealogists then get involved. They build out family trees based on these genetic connections, trying to pinpoint someone who could be the source of the unknown DNA.
That sounds like a massive amount of work.
It is. It involves careful triangulation, lots of traditional detective work, too. And this is crucial. Anyone identified through IFGG must then have their DNA tested using the standard STR method.
Ah, so the IFTG points the way, but you still need that direct STR comparison to confirm.
Absolutely. It's a confirmatory step. And generally, IFGG is seen as a method of, let's say, last resort when other DNA techniques haven't yielded a lead.
Okay, this is where it starts to get really interesting for our question. We hear DNA evidence and often think it's infallible, like absolute proof. But is it? Are there, vulnerabilities or potential problems?
That's a key point. While the underlying science, the core principles of DNA identification, are widely accepted and very sound, its reliability in the real world, that really hinges on doing everything correctly.
Meaning lab procedures.
Exactly. Proper sample handling, rigorous analysis, validated methods. There were some important early cases, like Castro back in the late 80s.
Castro.
Yeah. The court questioned whether the DNA evidence was admissible because there were concerns about the specific lab's procedures and how they interpreted the results. It wasn't about DNA itself being flawed, but about how it was done.
And you mentioned another one, Schwartz.
Right, Schwartz in the early 90s. Similar issue. DNA evidence from a private lab was ruled inadmissible because the court found the lab hadn't followed accepted scientific standards or been transparent enough.
So it's less about the DNA molecule itself and more about the human processes surrounding it. Collection, testing, interpretation.
That's a great way to put it. And the quality of the expert testimony explaining it in court is also absolutely critical. It's complex stuff.
Is there a difference between DNA proving someone didn't do it versus proving they did?
Yes, generally, DNA evidence that excludes someone saying this DNA definitely didn't come from suspect X is considered more straightforward and presumptively admissible.
Because it's a definite no.
Right. Inclusion evidence saying this DNA is consistent with suspect Y relies on statistics. How likely is it that someone else would share this profile? So it's powerful, but it's about probability, not absolute certainty in the same way exclusion is.
Have there been actual cases where someone was convicted, maybe based partly on what seemed like solid evidence, but DNA later showed they were innocent?
Oh, absolutely. The National Institute of Justice published a really significant report. Convicted by juries, exonerated by science. It looked at, I think, 28 cases initially.
28 people wrongly convicted.
Yes, convicted and later proven innocent through post-conviction DNA testing. And what's really telling is why they were convicted in the first place.
What was the evidence?
Often it was things like eyewitness testimony, which we now know can be incredibly unreliable, or older forensic methods like microscopic hair analysis, which just aren't as precise or definitive as DNA.
So in those instances, DNA didn't fail. It actually revealed the failures of other types of evidence.
That's a crucial distinction, yes. Think about Kirk Bloodsworth, convicted mainly based on eyewitness ID, DNA testing years later, exonerated him completely.
Oh.
Or Ronald Cotton, same story, eyewitness accounts were key to his conviction. DNA proved his innocence and identified the actual perpetrator. Dale Bruson, victim ID, hair analysis DNA excluded him. It shows how DNA can be this objective check on other, more fallible evidence.
Were there cases where the lab work itself, not just eyewitnesses, was questioned?
The report did mention cases where alleged government misconduct or errors, like faulty lab tests or misleading expert testimony, played a role. The Honecker and Kotler cases were cited as examples where issues arose.
It's a sobering reminder that the system isn't perfect, even with science involved. I think I read somewhere that even the FBI's own data points to this possibility of initial misidentification.
That's correct. The FBI looked at sexual assault cases where they performed DNA testing. In about 25% of those cases, the primary suspect, the person investigators were focused on, was actually excluded by the DNA results.
A quarter? That's pretty significant.
It is. It shows that even today, DNA testing frequently clears people who might otherwise have remained suspects based on other, less conclusive evidence.
So even though DNA is incredibly powerful, we still need to be aware of potential pitfalls or challenges.
Absolutely. Even when the science is solid, practical issues come up. Samples can degrade over time or due to environmental factors.
Without enough DNA, maybe.
Right. Sometimes there's just insufficient DNA to get a full profile. Or you might have mixed samples DNA from multiple people. Interpreting those can be very complex, requiring sophisticated statistical analysis.
And lab errors. Can they happen?
While labs have strict protocols to minimize errors, the possibility, however small, can't be completely ignored. Contamination, mislabeling, humans are involved. But generally, the sources suggest the underlying scientific principles of DNA analysis itself are reliable. The issues tend to arise more in the application and interpretation.
Okay, so we've covered how DNA is used, its general reliability, and some inherent challenges. Let's circle back to the core question about potential improper use or interpretation. How might that actually happen, connecting back to the concerns raised by cases like Reed and Kohberger?
Well, one major area is the risk of over-reliance on expert testimony. DNA evidence is highly technical, right?
Definitely not something the average person understands deeply.
Exactly. So there's a danger that a jury or even a judge might give undue weight to an expert's opinion simply because it sounds so scientific and complex, maybe without fully grasping the nuances, the statistical probabilities, or any limitations in the findings.
Like the CSI effect, but maybe more subtle.
Sort of, yeah. It really highlights how crucial it is for everyone in the legal system, prosecutors, defense attorneys, judges, and importantly, juries, to have adequate training or resources to understand and critically evaluate DNA evidence. They need to be able to ask the right questions.
It sounds like the legal system needs to become almost bilingual fluent in both law and the basics of forensic science.
That's a good analogy. Another potential issue is related to access and process. There have been instances where prosecutors might have, say, resisted requests for post-conviction DNA testing.
Even when it could prove innocence.
Or situations where, even after initial DNA results seemed to exclude a defendant, authorities continued to hold them while maybe trying to test other theories or individuals. The Kotler and Honecker cases were mentioned again in this context. Defendants were held even after tests pointed away from them, while the state explored other possibilities.
Which feels problematic from a justice standpoint.
It raises serious questions, certainly.
So, bringing this all together, Does the history we've discussed and these potential issues suggest that DNA could be used improperly in cases like Karen Reed's or Brian Kohberger's?
Based on everything we've looked at, the history of exonerations, the documented procedural issues in past cases, the potential for misinterpretation, it's clear that the potential for improper use or errors exists. DNA is powerful, but it's not magic.
The fact that DNA has overturned so many convictions based on other evidence kind of proves that the system can get it wrong, and DNA can be the corrective, but maybe it could also be part of the problem if misused.
That's the tension, isn't it? It's an incredibly valuable tool for finding truth, both guilt and innocence. But its very power means that if it's mishandled, misinterpreted, or presented misleadingly, the consequences can be severe.
Now, it's really important to stress our sources didn't give us the specific details on the DNA evidence in the actual Reed or Kohberger trials, did they?
No, absolutely not. The materials we reviewed provide the broader context how DNA works, its strengths, its weaknesses, historical examples of problems. They don't let us make any judgment whatsoever about the specifics of those ongoing cases.
So we can't say, based on this, whether DNA is being used improperly in those specific trials.
Correct. That would require examining the actual evidence presented, the specific lab reports, the expert testimony, the chain of custody, all of the details unique to those cases, which we don't have here.
So for someone following these high-profile trials, or any case involving DNA, what's the main takeaway message from this deep dive?
I think the key takeaway is that DNA analysis is truly revolutionary for criminal justice. It can lock onto the guilty and, just as importantly, free the innocent. However,
There's always a however.
right, its power doesn't make it immune to error or potential misuse. The history of wrongful convictions overturned by DNA is the starkest proof of that. It reminds us that no evidence, not even DNA, should be accepted without critical scrutiny of how it was obtained, analyzed, and interpreted.
So when we hear about DNA in the Reed or Kohberger cases, the real question isn't just, is there DNA, but how is it handled? How is it analyzed? How is it being explained?
Precisely. It boils down to the specifics. Was every protocol followed meticulously? Are the statistical interpretations sound and presented clearly? Are there potential issues like contamination or mixture complexity being addressed? Ensuring justice requires more than just having the science. It requires rigorous adherence to procedures and critical thinking about the results.
It means we need constant vigilance, really.
Exactly. Ongoing scrutiny of how this incredibly powerful technology is deployed in the justice system is essential.
This has been really illuminating and adds a whole layer of understanding when thinking about these cases. Thanks for breaking it down.
My pleasure. It's a complex but vital area.
And for you, the listener, maybe the thought to ponder is this. As our reliance on forensic technology like DNA grows ever deeper, how do we as a society build and maintain the safeguards needed to ensure its power is always used for justice and never against it? Something to keep thinking about.